All right. Welcome, folks. Let's see. We get situated real quick and we get to go. At least. Here. Okay. That'll work. All right. All right, let me share my screen and we'll get started. All right. There we go. So, let's give a brief recap, okay? Talk about metabolism. So, metabolism is eating food, right? The process of eating food. Sorry. A lot of stuff just happened. Let's try this again. Okay, so metabolism is the process of eating food, breaking it down, putting it in your body, and then using it, right? Burning energy, okay? So we talked about metabolism. Those three things that you can break food down into are carbs, fats, and proteins, right? And all of that ends up as energy, ATP eventually. So ATP is the currency of energy in the cells, right? Sorry, one a little bit more light. It's the currency of energy in the cells, okay? And there's some hormones that regulate that, which you should know. And it's explosions is what's happening. We're blowing up organic molecules, okay? And then we talked about glucose and carbs a little bit. Glycolysis results in a little bit of ATP, and then a lot of ATP down here. Right? So we are burning 2 ATP to end up with 4, to end up with 36, right? 36? Yeah, 36. All right. And then there's the Krebs cycle, aka the citric acid cycle. So know this. You're going to want to. All right. Practice it. Go home. You are at home. Stay home. Close yourself in your room and practice it. Okay. The electron transport chain happens, and that's carbohydrate metabolism. Okay. So keep this kind of in the back of your head a little bit because we can get to there from different places. Okay, so keep in mind that it's not always straight down to glucose. Sometimes we can kind of end up with different things and still end up with the same result. Okay, but the whole point of this is to produce ATP. Okay, because we use that for energy. All right, lipids. Let's go. Oh, that's not bad. Slideshow. From okay. All right, lipids. So lipids get broken down into triglycerides. Okay, the triglycerides are 3 lipids linked to a glycerol molecule. Okay, so if this is a glycerol right here, these are the 3 fatty acid chains that are linked to it. Okay, for the purpose of this class, these can be anything. Okay, these can be like I just had a brain fart. Like beta acids or what are the really important ones? beta carotenes. I don't know. But these can be like any fatty acids. Okay. Generally speaking, they're going to be like single bonded fatty acid chains. Okay, so very simple uh, carbon molecules, organic molecules. Okay, so like you can see this one is like a 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 fatty acid chain, 13, 14, 15, 16. So there's these are two different molecules. One is 12, one is 16. This one looks like it has 14. And you can link all that together as a triglyceride. Okay. Triglycerides don't always exist as triglycerides. Sometimes it's just fat uh, 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 molecules stacked on top of each other. But that's a pretty common way that they end up in the body. Okay, so there's saturated ones, which are all single bonds. There's unsaturated ones that have little double bonds that get kinked and stuff. So unsaturated fatty acids are things that are like fluid. So like those are your oils, right? So like think cooking oils or be an avocado or something like that, avocado oil. Things that are saturated or things more like butter, lard, stuff like that, that's solid. Okay. So so that's triglycerides. So that's fats. They can also exist in like monoglycerides. So eventually you'll just pull two fatty acid chains off here and you'll end up as a monoglyceride. Okay, so you have your 3-chain glycerol and then you have one fatty acid. Okay, pretty simple, but pretty straightforward. Like that's your building blocks. It's analogous to if glucose is the base brick, I don't know, the basic building block of sugars. Triglycerides and monoglycerides are the building blocks of fats. Okay, so this is actually a good picture. Okay, so when we are breaking down these fatty acid chains and these triglycerides, they don't like to be in solution, right? So if you had a meal, say, it's going to be some fat, some water, some other proteins and other stuff. And the chyme that goes into your stomach or comes out of your stomach is going to be kind of a sludge of all of that stuff. Okay. So not everything is going to be fat soluble, not everything is going to be water soluble. So how does your body kind of deal with that? It creates something called a chylomicron. So a chylomicron is a whole bunch of triglycerides, molecules, monoglycerides, other like fatty acid chains that ends up they kind of turn in on each other because they're fat soluble, and they put their water soluble side facing out, right? So these phospholipids, right, will kind of surround all of that. This is kind of like what's happening here is basically think of like if you have a cup of water and you drip like a drop of oil in it, it'll fall down and create like a little bubble that will eventually like float up to the top. But with that little bubble right there, that's basically a chylomicron. Okay. So this is a transport mechanism. Okay, so this is just how these fatty acids get transported in water solid, like in water adjacent things, right? So like plasma or getting into the intestine. Okay, so these are think of them as like suitcases. Okay, so you open the suitcase, you put all of that in, and you close the suitcase. You hand it to the TSA. Okay. Um, it carries it pretty much the whole way. Okay, so it goes from absorption all the way through to the liver, where it might get broken down and like repackaged, and then it carries those fatty acid chains all the way down to like its end target cell, right? So um, another way you can think of chylomicrons because you have these embedded apolipoproteins. So there's like little proteins that end up on the surface of these. So you'll have like your phospholipids, and then you'll have little proteins on the surface. Another word for chylomicrons is, hang on a second, image. High density lipoproteins. Here we go. So if I you have these options, okay? These are different kinds of chylomicrons, lipoproteins. Make that bigger. There we go. All right. So if this is a chylomicron, you might have triglycerides, and then C, I'm assuming is cholesterol. And then you have a chylomicron remnant, so that could be like a smaller version of it, and then a normal one. And then you also have VLDLs, IDLs, which is intermediate density lipoproteins, and then LDLs, and then VLDLs, and then HDLs. Right. So all of these kind of exist in the same sphere. So when we're talking like good fats versus bad fats, or good cholesterol versus bad cholesterol, or like the HDLs are great, you can have those, but the LDLs are bad, you can't have those. This is what we're talking about, okay? We don't measure like chylomicrons in your body, like that's just like not like the lab value kind of thing that we're looking for. What we look for when we look at your lipid panel, right, is we're looking at these VLDLs, your HDLs, your IDLs, and your cholesterol, okay? So we want to know how much of the fat that's moving through your bloodstream is a VLDL. Now, VLDLs are, let's say, bad. That's a bad terminology for it. They're normal, but they're bad because they're larger, right? So these are larger molecules that are swimming through your bloodstream, okay? So if you can repackage that into, say, 2 HDLs, that's good, right? So if you have a higher ratio of HDLs to LDLs or VLDLs, that's a great thing because you're packaging them more densely, and so they're moving through the bloodstream more safely, right? You're taking a big package that's bouncing around, making it smaller, and it's kind of going through more evenly, okay? That's a very basic way of explaining why HDLs are better than LDLs, right? So you want them to be higher density than lower density. Chylomicrons really kind of only exist in the portal system. Generally, they get repackaged in the liver and then into VLDLs, HDLs, and IDLs, all these LDLs. But if they do, they're very, very low numbers in the bloodstream. Okay. So when we look at a lipid panel, that's what we're looking for. Is we want to know what ratio of your blood is a lot of densely packed lipoproteins or loosely packed lipoproteins. Because if it's denser, it's more stable, and it's moving through your body at a more efficient rate. Right? It's not causing damage along the way. Okay. So that is how it works. All right. Oh, and the very last thing, all of that is kind of determined by these embedded apolipoproteins. Okay, I should have brought that back around. So these apolipoproteins determine whether something is like an HDL or VLDL, right? They think all the way back to the immune system, right? We've got the antigens that are kind of existing and being expressed on the outside of the cell. You can kind of think of these as their own little cells, right? And they're expressing antigens on the outside. They're apolipoproteins, but they're expressing these on the outside. That's holding this whole thing together, but then also giving the cells at the target organs something to recognize that with. Okay, so they'll see an HDL and they'll go, "Oh, I can take that in. I can pull that whole thing in, and I get a lot of dense protein for not a whole lot of effort." Versus a VLDL, it's I get less protein for more. So HDLs are good. Okay. Now, breaking down fatty acids. So, fatty acids are broken down during oxidation. Okay, so blowing crap up. Okay, triglycerides can be broken down into acetyl-CoA molecules and used for energy when glucose levels are low. Are we even talking about that? We are not. So, what I want you to know from this little area right here, there is something called beta oxidation, and that is how fats are burned in your body. Okay, that is a little more complex metabolism. Okay, so just know that for the purposes of this class, fatty acids can be broken down into acetyl-CoA molecules. Now, why is that good? It means that we can do more um, citric acid cycles, right? We've got more acetyl-CoA molecules. We can run those through the citric acid cycle more. And by default, we'll end up with more molecules that will go into the mitochondria and they'll end up in the electron transport chain. Okay, so it's good. We have precursors to the citric acid cycle, which is awesome. All right, so how does that happen? Sorry. All right, so this fatty acid, so we have a small chain fatty acid right here. It is moved into the cell. Okay, so from there it reacts with CoA prerequisite, right? And that creates CoA in a fatty acyl CoA. And then that produces something that reacts with carnitine, which is kind of like a amino acid, but not really. And then that creates fatty acyl carnitine. Okay, so you're just taking two molecules and you're putting them together. Okay, that fatty acyl carnitine enters the mitochondrial matrix. So alone, they can't go into the mitochondrial matrix. Together, they can move into the mitochondrial matrix. Okay, so that fatty acyl carnitine is then broken apart and it's turned into fatty acyl CoA, which is over here. Okay, so you're basically doing the same reaction to break it apart, and it's ending up as the same thing, but within the mitochondria. Okay. The answer to why does it do that? Why doesn't it just bring it into the cell? Is kind of complicated, and it's got to do with physics and stuff like that. Basically, it's easier to move that bigger molecule into the mitochondria than it is to move two individual molecules into the mitochondria. Go figure. We want to create reactions that happen spontaneously. So by doing that, you're creating potential for a spontaneous reaction. Okay. All right. All right. So from there, we've got the fatty acyl CoA, which is converted back into it was just converted into beta keto acyl CoA, which is split into acyl CoA and acetyl CoA. Okay. So those things are different. So acetyl. Let me see if I can show you. Here we go. Here's a Quizlet that I found online. Oh, I'm blocked. That's not nice. Okay, hang on. Here is something different. I don't like that one. Hold, please. I'll just show you this is what was on my Quizlet. All right, so acyl CoA is an R group. R is just like a random thing. Okay. R doesn't stand for random, but it's just a active group that's attached to a molecule, and that's attached to this CO double bond, and then there's a sulfur and a CoA. And remember, a CoA is a cyclic chain, I think. And then an acetyl CoA is just, instead of the R group, it's just got a carbon group attached to it. Okay, so a methyl group right there. So instead of having maybe potentially a long chain right there, it's just got a little methyl group. Okay. All right, so that's the difference between acetyl-CoA and acyl-CoA. Those two are different, okay? So when it's in the mitochondria, you have the acyl-CoA and acetyl-CoA. You'll take that fatty acyl-CoA. So remember, we've got like that R group right here. You're going to add FAD and FAD, and it's going to produce FADH2. And then from there, we're going to have a trans-eno an enoyl CoA. That's enol enoyl CoA. Trans-enoyl CoA. Okay. So from there, which is a different molecule again. All right. So from there, we're going to add water, right? And if we add water, we're going to add these H's, right? And then we're going to add FAD and FADH2 again, and that's going to produce beta keto acyl CoA. Okay, and then from there we can produce acyl CoA and acetyl CoA. Okay, now this acyl CoA, what happens to it? Mostly it just goes back out and comes over here. Okay, and it's recycled. Okay, so kind of the same thing that we talked about with like the electron transport chain. A lot of things enter the mitochondria, break apart, one will exit. They're using it as, think like transport proteins. So that's kind of how you can think of this acyl-CoA, right? And what we'll end up with is acetyl-CoA, which will enter the Krebs cycle, right? Because we're already in the mitochondrial matrix. So if we have free-floating acetyl-CoA that's the result of fatty acid breaking down, or if it's just the result of something else and it just ends up there, it doesn't really matter what it is. The existence of that acetyl-CoA is going to spontaneously produce a Krebs cycle. Okay. Chemistry. All right. Not going to make you memorize like the spontaneity formulas and stuff like that. That is just way too much. Okay. So we have acetyl-CoA. So say there's two of them right here. That will react. This next process is called ketogenesis. So we can actually go in reverse. Okay. So we're taking the acetyl-CoA. If there's too much of it, we can pull it away from the Krebs cycle and put it into ketogenesis pathway. Okay. This happens in the mitochondria of liver cells only. Okay. And what we're doing is we're storing that acetyl-CoA. Okay. So if glycogen is the storage. Glycogen is the storage form of glucose. Acetyl-CoA, the storage form of that is beta hydroxybutyrate. Okay, and we do that in the liver. So if we have too much buildup, your cells will push it out and be like, "Hey, liver, store this." Okay, so this will travel all the way to the liver. It reacts. Two of them will react into acetyl-CoA. So this is the acetyl, or this is the acyl. This is the acyl. This is the acetyl, because we've got this R group right here, and that will produce acetoacyl-CoA. A series of reactions. We end up with beta hydroxy, beta methylglutarol CoA, HMG-CoA. Just acetoacyl-CoA, HMG-CoA, acetoacetate, and then beta hydroxybutyrate. Okay. This one not quite as important as the Krebs cycle. I would rather you, if you're going to memorize one thing out of all of this, memorize the Krebs cycle. Okay, you just have to know it. And so this is called ketogenesis. This is different than ketosis. Okay, so we're going to come across a term called ketosis later, maybe. This is a different process. This is ketogenesis. Okay, we're creating ketone bodies that we can use to store acetyl-CoA. Okay, there's a lot of different ketone bodies, and they're all storage stuff. All right, so if we are beta hydroxybutyrate, we need to find a way to put that back into the Krebs cycle, right? Because maybe something has changed and we need to produce more energy now. So we're going to take that ketone and we're going to basically go in reverse, and then we can oxidize them into acetyl-CoA, which will end up back in the Krebs cycle. Okay. So again, like you don't even know, just kind of know that these are opposite mechanisms. Okay, they're kind of happening in reverse, and then we use this when glucose is limited. We need more acetyl CoA. Okay, so this is kind of that like the fat burning exercises, right? Like if we're using a lot of glucose in the first couple minutes of exercise, then we flip to a mechanism where we're using a lot of ketone oxidation to produce acetyl CoA and burn it in the Krebs cycle. Okay. But it's not like exclusively happening. This is just when we've been exercising for a long time and our glucose stores are starting to run out. Okay. All right. Lipid metabolism. Okay. So I don't think you need to know like all of this. The basic thing that I would take away from something like a slide like this is lipids. Lipids are long chain organic molecules. Okay, so there's not a whole lot of difference between like this and maybe if I were to slice it right here and put an OH group on it, that would end up being glucose, right? Because glucose is like a 6-chain carbon with alcohols on it, right? So if I were to add a couple alcohols on here and cut this into pieces, you might end up with a lot of glucose, right? Um, so they're basically good Legos. Like it's a good Lego set that you can kind of pull from and create a lot of different things from. Okay, so this monoglyceride, like by itself, can be used to create like fatty acids. All of this can happen in reverse. Malonyl CoA, which is part of the Krebs cycle, acetyl CoA, oxaloacetate, malic acid, pyruvate. So all of these things should be familiar to you. They're all part of glycolysis in the Krebs cycle, and it can all start from monoglycerides. Okay, so we can work backwards through all this and we can create whatever building blocks we need. So if we find that we don't have enough oxaloacetate to continue one of our metabolic processes, we can take fatty acids and we can just flip them back into oxaloacetates, right? Like there's mechanisms in place that all of this stuff can be kind of converted to something else, okay? The thing I remember when it comes to like metabolism and really chemistry, these aren't special, right? These little C's, they're just carbon atoms, right? So you can use these carbon atoms for anything you need them to, right? They're not like dedicated to being lipids. You can work your way back through different processes, and you can have this carbon atom could be the same as this carbon atom, all right, through a series of reactions. Okay, so just kind of remember that a little bit. Right, proteins. Any questions about lipid metabolism? I'll give you a minute. Ari's not here today, so she can't speak for everybody. So I need you guys to speak up. All right. Let's see. Click, click. Okay. All right, proteins. Let's talk proteins. So this happens kind of along the whole way. Proteins are a little bit more complicated than carbs and fats, right? So it naturally they're going to take a little bit longer to digest. Okay, we're going to need to employ a couple different mechanisms. So like we use the stomach acid to kind of denature the proteins. We use the agitation and the mechanical breakdown portions to kind of break them down. But then there's also like other hormones that we need to integrate into the digestive process to really reduce proteins. And the reason is, if you remember kind of what glucose looks like, Here, this is a protein. This is hemoglobin, probably. This is what proteins look like. But they're wildly complicated. They're massive, massive molecules. There's areas that are hydrophilic. There's areas that are hydrophobic. So like, it's not like you can just throw a bunch of water at them and they'll dissolve. Like you've got to dissolve part of it, and then another part of it. You've got to denature it. You've got to unfold them or unwind them. Like they do this crazy stuff where they kind of like they fold. There's beta sheets and there's alpha helices. So there's kind of like a lot going on when it comes to proteins. They create big long chains. So you have to kind of like integrate a lot more work into it. Okay. So for that reason, they move all the way through the digestive system. Okay. So they start up here, breaking down in proteolysis, which is basically just breaking down proteins through hydrochloric acid and other hormones. So pepsin is part of that. And then they move into the small intestine. This area of the small intestine, who wants to guess at what that's called? You already know it. It's the first stop on the small intestine. We've talked about it. Ah, come on. You can guess it. Duodenum. There we go. Yes, it's the duodenum. So this right here is this. This is the duodenum. Okay. So there is a little hole right here called an ampulla, right? And that links the pancreas and the duodenum. Okay. So the intestinal cells will secrete secretin and CCK. Those are hormones that break down proteins. And then the acinar cells of the pancreas will secrete trypsin, chymotrypsin, elastase, and sodium bicarb. And all of those things will be used to break down proteins as well. Okay. So the pancreas is not just important for like glucose control and diabetes and whatever. You're also secreting a lot of stuff that digests the proteins. Okay. And all of this happens in the duodenum. Why? Because right here is where the pancreas lies. It's posterior to the stomach, and it's right there, right? So there's a little hole right there, and it just squeezes a little bit of juice in there, and it kind of mixes up, okay? That hole is the ampulla of... Let's see. I think I can bile ducts. Yes, it is. It is this one. Here's a good picture. So there is a ampulla of Vader, which is right here, which is controlled by a sphincter, which is called the sphincter of Oddi. And there, the pancreas, the pancreatic duct, and the common bile duct meet. So your bile gets secreted into the duodenum through here, and so does your pancreas. It secretes all of this stuff through here, and that happens through the ampulla of Vader. I know what you're thinking. Why is this named after two different people? Well, they got in a fight about it, and they couldn't decide. So one got to call the outside of it the ampulla, including amphitheater, and one got to name the sphincter. Go figure. White man could not figure it out amongst themselves. All right. So you're gonna secrete stuff into the duodenum, right? What happens there? Let's talk about the energetics of amino acids, okay? So those can be broken down. So we've got proteins, they get broken down into amino acids, and all of those amino acids can be broken down into precursors for the Krebs cycle, okay? So there are 20 amino acids. Do you need to know them? I'm going to say no. I'm going to say you do not need to memorize a... Hold, please. I can't remember if I saw it on the test. If you need to memorize the amino acids, I will tell you in an announcement. Otherwise, assume you do not need to memorize the amino acids, okay? There are 20 of them. There are essential amino acids, which means that your body can not make them itself. And there are non-essential amino acids, which means that I don't care. I don't need to eat something with this amino acid. I can make it myself, okay? So those can be broken down as precursors for glycolysis. So these are kind of the areas where these get plugged in, right? So like if you start glycolysis and you end up with pyruvate, you're going to need alanine, cysteine, glycine, serine, and threonine. And then as you kind of move through here, if you're ketone oxidizing, right, and producing that CoA, you're going to need these amino acids. And then like at different steps in the Krebs cycle, you'll need different amino acids. And some of these are to produce those, like some of them are like precursors. So like alpha ketoglutarate is glutamine and arginine, I think, and they're linked together, and it's they produce a molecule called alpha ketoglutarate. Okay, so you do need them to kind of create molecules, but you also need them as like transport molecules. So there's something called the arginine and tyrosine create like an opposite pump effect, right? So that they can bring different cell molecules into the mitochondria. And then the same thing with like aspartic acid and asparagine. Those are basically the same thing with different charges. And you like go in with aspartic acid, you come out asparagine, and you end up with oxaloacetate. Okay, so they get plugged in at random points during the Krebs cycle. I'm going to say you don't need to know like where exactly they get plugged in. The most you might need to know about them is that some of them are essential and some of them are not essential. Okay, I don't think you need to memorize all 20. Right? I reserve the right to post an announcement telling you that you do. If I do, it's on the test. Is that fair enough? People are telling me that I'm not clear enough on what's on the test. I feel like that's as clear as I can get. All right, urea cycle. Urea is a byproduct, okay? So ammonia is a byproduct of the urea cycle to produce urea that's eliminated through the kidneys, okay? So the reason we have ammonia and urea is because of the byproducts of the citric acid cycle, okay? So if we take like CO2, for instance, and we run all the way down here, and we end up over here with, we'll end up with ornithine to L-citrulline. Citrulline becomes fumarate, which eventually leaves, and then we can kind of produce different, like arginine, right? That's an amino acid, so that can fall out of this and go do something else, right? But we end up with urea because it pulls those nitrogens off, okay? And we need to get rid of that, all right? That's basically what's happening with the urea cycle. Again, I'm going to say you probably don't need to like memorize the urea cycle. It's not super, I mean, it's important, but again, if you're going to memorize one mechanism from this chapter, I cannot stress it enough. It's got to be the Krebs cycle, okay? Know what the other ones do, know the Krebs cycle in detail, okay? Fun fact. Who wants a fun fact? Fun fact break. I studied, oh please. Sorry, my kid opened the door and said, I'm going to go poop. And then my dog choked on food at the same time, which was super fun. All right. What's I even saying? Memorize the Krebs cycle. The urea cycle. Oh, yeah. Fun fact. L-citrulline is a amino acid-ish precursor, and I studied that in college. And it occurs naturally in watermelon, and there is some studies that link L-citrulline to lowered blood pressure. So I was part of a study where I was a subject and also a researcher, which I think is probably a conflict of interest. But we were studying L-citrulline, so I was taking these massive amounts of L-citrulline supplementations, and then we were like testing blood pressure and like blood pressure dynamometers and stuff like that. It was really fun. But yeah, L-citrulline, if you eat watermelon, you end up with a little bit lower blood pressure. It was uh, enough to be like minorly significant in your diet. So, just fun fact: if you eat a lot of watermelon, you'll have lower blood pressure. There's a lot of other people in the background. All right, catabolic and anabolic pathways. So, um, what I would want you to take away from this is that all of these processes that we've talked about are all connected. Okay. So know that you can take glucose and you can end up all the way over here as a triglyceride, right? Know that as a triglyceride, you can end up all the way over here as something in the Krebs cycle. Okay. And from there, you can go through the electron transport chain. So, can glucogenic amino acids end up as energy in ATP? Yes, absolutely, they can because of this slide right here. Okay, all of these are connected. When I took metabolism in undergrad, I took over a library whiteboard, and I did like eating food, and then I drew the system, and then I drew out all the different pathways, and it was super fun. And I didn't sleep that night. It took me 12 hours to trace all of these pathways, and I still haven't found that text. It's very complicated. There's a lot of them. Just know that you only need to know a couple of them. And the most important one is the Krebs cycle. Have I said that yet? I don't know if I've said that yet. All right. Energy things. I think I was in stuff. All right, we're gonna stop here. We'll end it a little bit early, and we'll stop here. And I will pause for questions if you guys have any. Otherwise, I'll see you in live. We'll talk about something completely different. Okay? Capiche? Okay.